Hi everyone, I'm Professor Sarah Rankin and I would like to welcome you to Imperial COVID-19 lockdown lessons. So today is lesson 8 and we're going to be hearing from Dr Blerina Ahmetaj-Shala and some of our current students studying on the Masters in Genes, Drugs and Stem Cells. So they're going to be telling us about some of the medicines that are being used to treat patients who have been hospitalized with COVID-19. Please, obviously, as always, be free to post any questions you have in the chat on YouTube. Margarita is here as always, and she will be looking out for your questions to ask at the end. And you can ask about careers or you can ask about the science. As always, please have mentimeter.com open on a web page ready for the quiz question. For those of you who haven't used it before, you don't need to register or sign in for anything. You just go to menti.com and you enter a code. So today's code is going to be, again, it will be shown in the chat on YouTube when we need it, but it's 701407. So, Blerina, she's going to kick off for today. Now, I know, Blerina, you've got a degree. Your degree was in pharmaceutical sciences, and you're going to be telling us about what exactly pharmacology is and the difference between pharmacology and pharmacy, I think. So take it away, Blerina.
Great. Hi, everyone. Thank you for joining us today. I hope you enjoyed today's session. So today, as Sarah said, I'll be telling you about what medicines could be used to treat COVID-19 patients. And while we present to you some slides, if you could put in the chat whatever you think of potential medicines that could be used to treat COVID-19, that would be great. So as Sarah said, I'm Blerina. I'll be presenting with Yanis, Gloria and Alex today on this. So just a bit about my background and how I came into pharmacology and where I am today. So I'm going to break it up into 3 sections. So my school, my university and post-university. So I did my A-levels in central London. So that's where I've grown up back many years ago in 2003. So I did quite well. I could have done probably a bit better. But it was quite good. And then I decided to do a degree in something that was a combination between biology and chemistry. I did really like chemistry, but I couldn't do pure chemistry because I thought it would just be too challenging. It kind of frightened me away. So I thought I'll do something which encompasses both biology and chemistry. So I went and studied pharmaceutical science at Kingston University. And then I made lots of friends, I enjoyed it there, and I went on and continued to do analytical chemistry, so kind of looking more into drug testing, how we can test drugs and different types of drug testings that we can do. But just to make it a bit more fun, because pure chemistry, like I said, was just too much for me, I decided to do the degree with management studies, so learning more about how we can manage people and organizations and a bit of finance too. And then there was a PhD being advertised at Kingston, and it was in combination with the MRC Clinical Sciences Centre based at Imperial, and the PhD was in pharmacology. And I was really interested because I really enjoyed my project that I did for nearly 2 months in the summertime, but I was a bit worried because I hadn't really had all that intense pharmacology background learning that you would probably expect. But actually, when I spoke to my supervisors at the time and a lot of professors at the university, they said, yeah, why not? You can change fields. You can go into study pharmacology, and chemistry, biology, physics, maths, they all kind of interlink with one another. So that made me a bit more comfortable. And then I went on to study pharmacology PhD. I really enjoyed it. And then I've kind of been in that field ever since. So here I am today. I'm at Imperial. I've been working there since 2013 at the National Heart and Lung Institute. Initially I was a postdoc, so now I've been getting my own funds, and I'm kind of studying in my own area, trying to develop an independent group and doing some lecturing. So this is me, this is my background, how I came to be here. But today I'm going to talk to you about pharmacology and what is pharmacology. So if you go into Boots, I'm sure you're going to be bombarded with all different types of medicines. As you can see here, there's many different types of drug. And what is a drug? A drug is basically a chemical substance which we know the structure of, and if you give it to a living organism, so that could be a human, an animal, it could be anything that is a living organism, it produces a biological effect. And there's three different types of drugs. So you have drugs which are naturally present in the body. So an example of this is adrenaline. So obviously I'm standing here presenting to lots of you. It's a bit frightening probably. There's a lot of adrenaline pumping in my body, which is probably increasing my heart rate. So that would be adrenaline, and that's naturally occurring. So there's also other types of drugs which are derived from natural products. So these are drugs that could be derived, for example, from herbs, and you may have heard of herbal medicine. And an example is the willow tree. And if you go back many years ago, so to around 400 BC, where Hippocrates actually found that if you take leaves from the willow tree, it can relieve pain. So actually, you know, herbs and using natural products has been around for many centuries. And the last type of drug is the synthetic man-made drug. And these are drugs that are designed where you basically combine different compounds together, and then you make these in a laboratory essentially. And an example of the first ever man-made drug is aspirin. And so aspirin was first made in 1899 by the pharmaceutical company Bayer. And obviously now we use aspirin, and you know, you could probably say, I don't know anyone who has not had an aspirin tablet ever. So it's a really common drug and we use it every day. And so there's been a lot of work in these type of drugs, and I'm going to ask you if you could pop down in there who knows who won the Nobel Prize for discovering the pathway of aspirin, and then we can talk about that at the end. So these are the three different types of drugs. So you know, there's three types, and what pharmacology is the study of these drugs and how they affect our bodies. And why is it important? Well, it's important because it's everywhere around us. Sometimes you may not even think about it, but for example, if you went to the dentist and you had an injection, so he's giving you something that's going to numb your gums, for example, that's having an effect on your body. Also, just as a theory, if you're having a cup of coffee, there's caffeine present in there, so that's also going to affect you know, what happens in your body. So essentially pharmacology is out there and we may not even be aware of it. And why is it important? Well, there are many different reasons why pharmacology is important. It's important because there's many diseases out there, sadly, and we need to try and treat these diseases. A lot of people are dying from diseases like cancer, and we need to try and discover new medicines to basically fight these diseases and reduce the number of deaths. It's also important because we need to try and make medicines more effective. So while some medicines are good, they could be better. So maybe designing and developing new formulations of medicines is another reason why pharmacology is important. Also, we know that whilst some medicines are good, they also have side effects. So an example which you may be aware of is ibuprofen. So again, like aspirin, ibuprofen is very good. It can help alleviate pain, but ibuprofen comes with side effects as well. So for one example, side effects is that it can cause GI, so gastrointestinal side effects. If you take it for a long time, it can cause gastrointestinal bleeding. And there's also some potential people who can have an increased chance of a heart attack and stroke. And our group is kind of working in that area as well. So these are things that pharmacology can help target. So looking at how we can reduce side effects of drugs. Another reason is the fact that, you know, some people respond differently to medicines. And why is this? So learning more about why certain people respond differently to drugs is something that pharmacology can allow us to do this. And so just down here on the bottom, I put the British Pharmacological Society logo. And this is basically a network of people who are interested in pharmacology. So they could be people like myself, they could be people like yourselves, undergraduate, postgraduate students, world-leading professors who all have an interest in pharmacology, and they join the society and they meet once a year and they present their work, and we feedback to each other, and feedback is very important. And if you're interested in learning more about the British Pharmacological Society, there's the website is down below, and they have lots of useful links which you may find to be quite good and quite useful for you at this stage. So if you have some time, pop on that website and have a look. So this is a bit of a busy slide, but just to show you some of the examples of the different drugs that pharmacologists are trying to develop to treat a wide range of different diseases. So I've mentioned cancer. So a lot of cancers, there's no cures. We need to try and develop drugs for that. HIV, it's been around for a while, but still we're not at the stage where we can really fully treat HIV. Other examples include multiple sclerosis, neurological conditions such as dementia, Parkinson's and Alzheimer's. Also asthma. I guess we live in London, very busy place, increasing chances maybe of getting asthma and other respiratory conditions. So there's real need to really develop new drugs, and pharmacology is really the pivotal point of it, where we can use our understanding of biology, chemistry, and really understand how drugs work and how we can make drugs better. So what drugs currently exist out there in terms of if we look around the world? So this slide shows the world's most important drugs, and it shows when they were first kind of discovered. So we can see here, if we just look at penicillin, so this was discovered in the UK in 1928. And to be honest, I know for myself, I don't know what I would do if there was no penicillin or antibiotics. So penicillin was the first antibiotic to be discovered, and actually, as that statement says, that 75% of the world's population would not be alive if we did not have penicillin. So this is a big statement, and this just goes to show you how important pharmacology is, and drug testing and developing new medicines. Another example is smallpox. So you may not be aware of smallpox now, thank God, but that's all because of these studies that happened many years ago. So back in 1876, the first vaccine was first developed, and that was on smallpox. And thanks to this vaccine, basically smallpox has been completely eradicated now. I'm not going to go into all of them, but again, we have insulin here, so that's used to treat diabetes. That was discovered back in 1920. There's a lot of different other drugs like morphine. We also have aspirin, so I mentioned aspirin again, so I hope you're all typing away at who discovered the pathway of how aspirin works, and won the Nobel lecturer. So aspirin again is a very important discovery, and there's 40,000 tons of aspirin that are consumed each year. So this just goes to show you how important pharmacology is. And why is pharmacology important in terms of COVID? How can it help us? Well, obviously now we're kind of easing lockdown, and things are starting to get more back to normal reality and how we lived our life before this. But actually, if you look at this graph here, you can see, so this is a graph from the Worldometer's website. It's quite a good website actually, because it's like a live feed. So you can see how things and numbers are changing literally in seconds daily. And we can see here that the number of daily cases of confirmed COVID are increasing, although we're still beginning to ease our lockdown. So if you just go back a couple of days ago, there were 170,000 new cases and 5,000 deaths. So although COVID is we feel like it's kind of going away slightly, it's still with us very much so. And although a lot of people are recovering, a significant proportion of people are dying. And actually that's a 3% chance of actually dying from this. So we really need to try and develop a drug or a vaccine, and we need to do it quickly so we can help those people that need it the most. And this is where pharmacology can come in. It can really help us learn more about what drugs are out there. Can we make a new drug? Can we use drugs that exist? And so my colleagues will tell you more about that later. But in the meantime, if you could just go on to menti.com, so Sarah told you about that, and if you put on the code 71407, And if you can answer the question, how long do you think it takes to develop a drug, and how much do you think that it costs? So is it A, 3 to 5 years and 10 million pounds? Is it B, 6 to 9 years and 500 million pounds? Is it C, 10 to 15 years and 2 billion pounds? Or is it D, 16 to 20 years and 10 billion pounds? So we're going to tell you the answer for that shortly, but after that's pretty much me done. So after this, we have Yanis telling us more about the challenges that we face with making new drugs. So Yanis, feel free to take it away.
Hi everyone. So I've been just talking you through my background, my pharmacology journey. So I've started with medical studies because obviously I was interested in health studies and wanted to know more about how body functions. So it was a fail, but I bounced back and started biomedical sciences. And through my excellent grades, I had the opportunity to go to Imperial 2 years ago as an Erasmus student to finish my degree. And I totally loved it. So I decided to apply for a master's, which is called Genes, Drug and Stem Cells, which studies regenerative medicine, gene therapy, and pharmacology applied to novel drugs. So why is it difficult to make new drugs? Let's take a look at this cartoon, and it summarizes really well the field in the pharmaceutical field today. So before you had this little tree which shows that you could pick up easily the drugs, as Blerina just told you about penicillin, for example, because the reason is that because the technology at that time and the regulations were not strict enough, like it is today. So the leftovers we have now are like leftovers. There are plenty to find more, but there are all these rare diseases that are not that nobody were looking into them really. So now they are pushing the companies to go and find new drugs, new vaccines, new therapies to tackle these diseases. Now, oh yeah, you could still answer the question on Mentimeter. I'm gonna give you the answer in the next slide. So
would it be possible to see which what the people have thought?
Oh yeah, sure.
To share the Mentimeter slide?
Yeah, go on.
So I don't know. Okay, well maybe you can share the answer, Yanis, if that's.
Yeah, that doesn't work.
Yeah. So what's the answer?
So the answer is C, 10 to 15 years and 2 billion pounds. So let me show you how. So this is the drug development process and how a drug is discovered and developed from scratch. So after choosing your target, let's say a receptor on a specific molecule, you perform high-throughput screening on libraries of containing thousands and millions of chemicals, basically. And you test them in the lab, as the stage 1 just shows on the up there. So you have around 10,000 compounds to test, and using robotics and data processing, you screen that and test them. So now you can go onto stage 2, where you have around 250 compounds. So because you can't test it directly on humans because it's unethical, you want to use murine or canine animal models. So what do is you test different dosage at different times in order to see what's the best route to give your drug. Is it injectable? Is it given orally? How much time do you have to give it? 1, 2, 3 times? So all this, like the toxicity also, all this is tested during this stage. After that, you have around 5 compounds that you think that they can really work on humans. So then you just drop a patent application and start the clinical trials. So the phase 1 is called safety, because you will take just healthy volunteers and test how well it works on human. So you will give raising dose to people and see how they react, how their body organs react to the drug you're giving. Once that is validated, you go on to phase 2, which is efficacy. And this is called efficacy because you want to see the real effect of the drug on the body on a larger scale. So here you have a group of healthy volunteers and a group of patients who are attained by the disease you are targeting. And obviously, I'd say the gold standard clinical trial would be randomized, blind, and oh, I forgot. Alex will tell you about that just later. But anyway, the second phase, so efficacy, just assesses how well your body responds to the drug. And then once that is validated, you can go in a larger cohort with more than 1,000 people, which is the larger scale. And you also have healthy volunteers and a bigger cohort of patients. And the last word was placebo control. That's so once you've done all this, you have to get the approval from the Food and Drug Administration in the US, or the EMA, so European Medical Agency in Europe, in order to be accepted and approved. So then you can sell your drug to the population. So yeah, that's around 10 to 15 years and around $2 billion pounds, sorry. So now, why is it difficult to make new drugs? Here you can see the investment in drug development by the pharma companies, and how much approved drugs there are. So during the past 25 years, you can see that the number of approved drugs didn't raise really, and whereas like the investment raised steadily. So this is due to the technological advances we have, the regulatory rules which are more stringent today, and also that's a thing that came out during these 30 years. This is called drug repurposing, and Gloria will talk to you about it.
Great. So thank you, Yanis. Yeah, so Gloria is now going to talk to us, and Gloria Opera is going to be telling us about this drug repurposing, because what you've gathered from Yanis there is that if we wanted to create a new drug to treat COVID, you know, it's going to take at least 10 years. And so therefore, it's really not possible, or if we did it, we wouldn't be able to treat people. And we have people obviously in the hospital that have got COVID-19. We need to know what medicines to be giving them now. And so Gloria is going to be telling us about how we do this thing, which is called repurposing drugs. But before that, I think, Gloria, you're going to just tell us about your sort of career path to date. I understand you're interested in doing a PhD in genetics, so tell me what has led to that sort of decision and your career path.
Okay, so hi everyone, I'm Gloria, and I've always been interested in science. So I did my A-levels were all in all the sciences, and then I decided to do biomedical science at the University of Hertfordshire, only because I felt like that was a broad enough course for me to really explore the subject and to really find out the path that I wanted to take and what I really liked. So after doing that, I decided to do a master's at Imperial College, which is what I'm doing now. And I chose to study in the pharmacology stream because that is the one that I found the most interesting during the course. So, however, going on from this, I do want to study a PhD in genetics, only because I find genetics really, really interesting. And I do feel like the knowledge I have learned in pharmacology as well will help in that way as well. So one thing that I like about science is that you don't have to particularly stay in a particular subject or particular topic, because having the previous knowledge I have from biomedical sciences has allowed me to be able to explore different areas of science as well. So that was what led to my decision right up to today. So I'm going to be talking to you about drug repurposing. So what Yannis has said is he has talked about making drugs from scratch. I'm going to be talking about using drugs that may have been already made but not used. So drug repurposing is, at its most basic level, it is linked to recycling. So basically, it's taking a drug that is intended to treat one patient group and using it to treat another completely different group of patients. So the drugs tested for repurposing include those that may have already been approved to treat a different disease, such as inexpensive drugs. It doesn't have to be an expensive drug, maybe like aspirin or any drug like that, along with drugs that have been put through millions of dollars worth and years worth of testing, but they have been shelved and not gotten FDA approval. So it's like a race, but in this race of drug repurposing, you're closer to the finish line rather than right at the start, which Janice was talking about. So because these drugs have already extensively been studied and they've been tested in people as well, you can skip some of these clinical trials and even phase 1 maybe, which saves time and lets you get them to treatment the patients faster. So why do you repurpose drugs? So instead of wasting all this money and all this time in drugs that may have been shelved or not approved by the FDA, you can repurpose them to reduce costs, and it takes a shorter development time because, as I already explained, that they have already underwent testing in humans to prove that they are safe to be used in humans as well. And we know that the drugs are already known to act on a relevant target as well, because and science backed this up because of the fact that some drugs act on off targets. So they have effects on other things. So you may take one drug for one specific reason, but that drug can influence a whole other lot of stuff happening in your body. And this is why repurposing drugs is actually a thing, because drugs can influence more than one thing. So we know that it's safe also because it has been tested previously in patients as well, in trials. So here's the ideal process. So compared to Yannis's explanation of the process taking up to 10 to 15 years, maximum a repurposed drug will probably take up to 8 years, if that. This only depends on how far the drug went before it got stopped to be used. So the first step you have is the compound identification. This is where you choose your candidate drug. You choose the drug that you want to repurpose. You analyze it. This takes about 1 to 2 years, and then you have to obtain a license for the drug. Because some drugs are already licensed to some companies, you want to ensure that you have the license to use this drug and to market this drug for something else. Because if not, you could be in breach of copyright infringement and you could get into a lot of trouble with the law. So once you've obtained this license, it can take up to 2 years to get a license. And then you enter the development stage. Now, this stage can vary depending on how far the drug went for its original idea of treatment. So you can start at either phase 1 or phase 2. Preclinical trials will have already been done, so you wouldn't have to do that at all, which saves a lot of time and a lot of money and a lot of years. So this is just to make sure that the drug is still safe, ensure the safety of the drug. And so after this, and has been developed and been proven to work in the treatment, in the way you want to repurpose it, then it goes to the FDA to be approved. So once this is approved, it's monitored by the FDA to make sure that it's safe for the public. So all in all, drug repositioning means that it's shorter time cost, shorter financial burden, smaller financial burden for drug development, and also you need lower R&D investment required as well. So another part of drug repositioning is something known as drug rescue. So it's similar in that some drugs may have failed in clinical trials, which means that they may not have been able to treat effectively what they were intended to treat in the first place. So because of this, you would have a huge financial loss because investors are always pumping money into drug development. But since it didn't work, people are going to lose their money, you're going to lose a lot of time, and also goodwill as well, because we know that some diseases, there are charities for them, and just these charities donate for research. So all that would have been lost. However, you can rescue a drug by conducting some analysis after the drug has been written off as probably not effective. So post hoc analysis just basically, it involves you looking at the results and looking for other things that may have happened that you were not looking for in the first place. So remember I talked about off-target effects. So off-target effects could have happened that you may not have noticed, but it could have worked on another pathway, but not the one you were intending to find. So once you have found this out, you realize that even if this drug was not effective for condition X, it can be effective for condition Y. And this is how a drug gets repurposed. So one of the most popular drugs that have been repurposed is sildenafil. Most of you may know it as Viagra. So Viagra was intentionally made to treat pulmonary hypertension. However, now it has been repurposed to treat erectile dysfunction. And this is because it acts the same way that both diseases kind of act the same way. It acts the same way as to increase the blood flow. And so because of this drug has been repurposed and now it's very, very popular. So a lot of drugs have been repurposed for different things. So now Alex is going to talk to us about potential drugs for the treatment of COVID-19.
Right, hi everyone. I will start the same as the others, is with a kind of brief synopsis of my pharmacological journey, if you will. So very quickly, I began my higher education up in North London, studying mathematics, biology, chemistry, and physics, which made for a relatively challenging combination, especially for someone who was not remotely kind of interested in listening or studying it at the time. So as I've noted there, I came very accustomed to failure due to my own negligence, which in hindsight was great for me, I think. I never planned to go to university, so I spent a year on building sites and labouring as a bike mechanic. But after a year, my brain was kind of yearning for some intellectual stimulation. So I've always been interested in how things work, and the body is a fine, complex system. So I chose a degree 2 in biomedical sciences, and it kept me seriously intrigued for the whole 3 years. I had the most amazing time, and you'll cover a vast expanse of content, including immunology, cancers, cellular mechanics, protein form and function. The list is extensive. However, pharmacology was a reoccurring topic that kind of tweaked my interest. So uncovering drug action and how these little white pills you take really work in the body. Anyway, that led me on to Imperial, onto this master's course, Novel Therapeutics, which contained everything I was really intrigued by. Not only the latest drug strategies, but futuristic treatments like gene therapy and stem cells. So it's pretty exciting stuff. And I'm especially fascinated by the kind of commercialization of a medicinal product, especially the legal aspect. So I'm searching for kind of jobs in patenting and IP currently. Anyway, onto the interesting stuff. So before you take a drug to the market, it needs to go through a vigorous series of testing to ensure it's effective and safe, as Yanis has mentioned previously. As you can imagine, there's a real need for this, especially for those of you familiar with the kind of thalidomide tragedy in the 1960s. So essentially, clinical trials are very simple. They compare the test treatment with a treatment currently used in an effort to prove what you've created is better than what's already here. And of course, to show that the side effects are not so horrendous that people will be actually able to take it and comply with the taking. So this website here, clinicaltrials.gov, is a searchable database of clinical trials conducted all around the world, and it gives you information on location, participation, why they're doing it. As you can see, as of yesterday, there was 2,371 studies going into COVID alone. So the effort is immense. It's incredibly large. Now, clinical trials aren't always conducted correctly. Now, what I mean by this is that there can be an element of bias, which often throws off the results and creates false results. This news line down here in the bottom right-hand corner, a couple of months ago, there was a lot of ranting and raving about a drug called hydroxychloroquine, and it's a drug mostly used for malaria. However, some results from a couple of trials led to Donald Trump making a speech about its effectiveness and claiming that he'd been in fact taking it for weeks. Anyhow, since then, larger, more reliable clinical trials have stated no beneficial effect of this drug at all. So you've got to be careful. So this is a brief slide. There are 3 simple elements which aim to eliminate bias on a clinical trial and hopefully eliminate these dodgy results. Number 1 is it needs to be controlled. This is really simple. This just means the clinical study must include a comparison group as well as your treated group. The comparison group will receive a placebo, another treatment, or perhaps no treatment at all. And it's important to realize that these 2 groups will be as similar as possible. I'm talking average age, similar genders, height, weight. And the more you control, the more accurate results are. Number 2 is blind. This means information that may influence participants in the trial is withheld until completion. So effectively, participants cannot know whether they are on the experimental drug or the old drug or the placebo. The best experiments will be described as double blind. So as you can see in the title at the bottom, this means that not only the participants don't know what they're receiving, but neither do the doctors giving it to them. And this really cuts out a lot of bias. Lastly, is randomization. This is super simple. This describes the process of randomly allocating subjects into different groups, and this is normally done via computer. So these are the 3 repurposed drugs I've selected to show you. The first 2 aim to reduce the body's immune response to the COVID-19, whilst the last one aims to target the virus directly. So the first in the green, if you cast your eyes over to the left, is dexamethasone. And lots of you might have heard this in the news quite recently. It's quite simply a corticosteroid, which aims to reduce inflammation. As many of you already know, a big issue with the COVID-19 is that the body tends to mount an overwhelming immune response to COVID, which results in a lot of damage. So dexamethasone, it's a commonly used steroid used to treat things like arthritis, allergic reactions, asthma, COPD. All these things involve a kind of chronic element of inflammation. So it's been around since the kind of the late 1950s, and it's thus, very cheap and in wide production. So it's been calculated that a COVID patient would require a little over £5 a day of this stuff to keep him alive. It's been dubbed the first life-saving drug for COVID-19. And it's been said if we'd been prescribing this alone from the beginning in the UK, that 5,000 lives would have been spared already. It's especially shown its effectiveness in ICU patients, reducing the amount of deaths on ventilator by 1/3 and 1/5 on oxygen. Right, next is tocilizumab, which is a different class of drug entirely. Although it aims to still dampen the immune response by binding something called IL-6, which is an immune modulator. Rather than being a small molecule drug, the drug itself is a large protein. It's essentially an engineered human antibody. And these are kind of the future of drugs. You can always tell a kind of humanized antibody in the title of the drug. The ending mab indicates this. So tocilizumab is used to treat autoimmune diseases such as rheumatoid arthritis, which results from kind of severe inflammation in synovial joints. And although, far more expensive than the dexamethasone, the price is on the way down. Functions targeting the cytokine storm, by binding one of these key elements. So last but not least is remdesivir, and like dexamethasone, it is a small molecule drug again, so not one of these large kind of antibody type things. Yet it aims to target the viral cycle rather than the body's response to the virus. Arguably a better method, maybe. What do you guys think? So it's a broad spectrum antiviral, meaning it's designed to be effective against a whole host of different viral infections. It seems it's kind of mainly effective against the RNA-based ones. It was originally designed to treat hepatitis C, which is the image you see on the left, and later during the Ebola epidemic, which is the kind of worm-like virion on the right, which were both rather unsuccessful, may I add. However, it seems we're having a little more luck with the COVID. A study released last month showed remdesivir treatment sped up the recovery by an average of 5 days. Controversially, though, The Lancet, which is among kind of the oldest and best known general medicinal journals, published a study claiming that it had significantly insignificant clinical benefits. However, this hasn't stopped its approval in the UK and emergency approval in the US, and at present it's being used to treat patients, although supplies are limited. Which one would you go for? Finally, I just wanted to give you kind of a flavour of alternative approaches, bearing in mind there is, 2,000 plus initiatives being tested as we speak. The figure here is a kind of simplified life cycle of the COVID virion. I've split it up into 3 main stages. As you can see at number 1, the virus binds this ACE2 receptor here on the host cell, gaining entry. Now, here could be a potential method of perhaps blocking the virus's entry. Post-entry at number 2, it releases its contents into the cell and begins harnessing host cell machinery in order to replicate. Now, this is where remdesivir acts to stop the virus, and what we discussed earlier. It effectively acts as an alternative nucleoside analog, which blocks transcriptional machinery. And then if you just cast your eyes over to number 5 and 6, the virus is then repackaged and buds out of the human cell. Now this process often requires enzymes called proteases to basically cut the tethers from the cell so these viruses can escape. Designing drugs to inhibit these enzymes has proven successful in anti-HIV drugs. So perhaps we could repurpose here, I'm unsure. Lastly, I just wanted to mention the kind of donor plasma program, as you can see on the right. This is a form of treatment occurring and being trialed all over the world, including the NHS right now. The premise is that plasma, the yellowish liquid in your blood, from someone who has recovered from the virus may save lives of individuals who are still ill. So essentially, the plasma from people who've had the COVID may contain these antibodies produced by their immune system. This plasma can then be transfused into patients who are struggling to produce their own antibodies. So it's essentially a kind of donation immune system fight injection. Anyway, there's currently 2 trials ongoing in the NHS, and these seem pretty exciting too. Anyway, that concludes what I've had to say, and thanks for all of you that's listened all the way till here.
Great. Thanks, Alex. That was really clear. That last thing about the transfer of plasma is essentially what you get with mothers that are breastfeeding, and you transfer the antibodies via milk. So that sort of, helps babies, very young babies, have immunity to lots of general pathogens before their immune systems have properly kicked in. So lots of things to pick up on there. One thing I'd like to mention, so Blerina, you mentioned the British Pharmacological Society, and I'd just like to say that on there is a section about for schools and careers, and they've got some really nice sort of materials talking about all the different alternate careers you can go into as a pharmacologist. And we heard today how, whereas Blerina is a sort of bench scientist, so she's doing experiments, whereas Alex is thinking of more going into what would be more a desk job, thinking about the patent and licensing process. So yeah, there are lots of different alternate careers, and I think picking up on these drugs, so the drugs that are being used, if you go back and remember the very first COVID-19 lockdown lesson we had with Professor Danny Altman, he was talking about that immune response to COVID. And now you can see why it's important to understand what that immune response actually is, because then we can use drugs that target different processes in it. And that won't cure you, but it will reduce the severity of the disease caused by the virus. So you're not actually preventing the virus as such, but you are preventing the subsequent disease. And yesterday we were hearing about the work done by Professor Peter Openshaw's group and others across the UK in coordinating the collection of patient samples. So they were collecting things like blood and small pieces of lung from people that were hospitalized with COVID-19. And that was really important, because then again they can analyze in greater detail exactly the immune response of people at different points in the cycle from infection. And again, this will give us as pharmacologists clues how to tackle the infection or the sequelae of the infection. And so in this way we can sort of refine treatments. And one of the things I think is really interesting, and I don't know if people have commented on it yet, but at the beginning of this pandemic, we were worried about that there were specific groups that were going to be susceptible. And one of the groups we thought would be very susceptible to the virus and actually the sort of negative effects were people with asthma. And actually, there doesn't seem to be much to show that these people are really have an increased susceptibility. And it might be because a lot of those patients are actually taking dexamethasone, that anti-inflammatory drug. So they might in some way actually be a bit more protected. So that's the sort of thing that will be interested, and we will be finding that out when we do further sort of interrogation of all the data that people like Peter Openshaw's groups are collecting, because one of the things we will know is what, clearly we have people that are having drugs for other treatments, say for hypertension or something, and then getting COVID. And so one of the other things that we're interested in as pharmacologists is what are called drug-drug interactions. And so it's really important, to ensure if you're taking one drug, is it then safe to take another drug? So, for example, we know if you've you consumed a lot of alcohol, then it's not safe to take paracetamol. And we know why that is, because of the pathways. So I was just wondering, I'm going to go over to Margarita now and to see if anybody's got questions. And I just wondered if anybody had got an answer. So one of the questions Blerina came up with was, there was somebody, actually a UK scientist, that got a Nobel Prize because he worked out how aspirin works. So most drugs work. There's two very common mechanisms by which drugs work. One is that they bind to a protein on the cell surface, and that protein is called a receptor. So they block the receptor. So if you think about it, it's quite often termed the sort of key and lock pathway. So normally you put a key in a lock and turn it, and that gives a response. When you have a drug, you basically like putting chewing gum or blue tack in that lock, and you prevent the key going in to give a response. So that's one mechanism by which drugs work. And the other mechanism by which they work, which was found for aspirin, is that they block the activity of an enzyme. So within your sort of GCSE and A level, you would have heard about enzymes. Enzymes obviously really important in catalyzing chemical responses. And by blocking enzyme activity, you can slow down a lot of biological processes and sometimes inhibit them. And so what was found with aspirin, or acetylsalicylic acid, as it's known, its sort of chemistry, is that it actually blocks an enzyme which is referred to as cyclooxygenase. So, and that enzyme is normally important in generating inflammatory mediators. So it stops the formation of these chemical substances which normally drive an inflammatory response. Right, so that's me. I should stop chatting and go straight over to Margarita for anybody that actually correctly identified that person, and have we got any more questions?
Yeah. Hi, Sarah. I'm really excited today.
Name of the professor was Professor John Bain, so he's the one who basically Sarah was talking about. And he used to work at Queen Mary's University, actually, which is quite interesting. It's quite close to us. So to think that this big discovery was made just a couple of stations down us is quite exciting.
Well, and also, I guess more importantly for us at Imperial, that Sir Alexander Fleming was based at Imperial at our St Mary's campus. So discovery of penicillin, which is undoubtedly saved like billions of lives over many years, was also done there. So yeah, Margarita.
Yeah, great. I would like to say that I'm really excited for today's lesson because I had the opportunity to meet my classmates. Alex, Gloria and Yanis are my classmates, so I haven't seen them for a while due to lockdown. So I'm really happy about it. And the question that came up has to do about studying and how to choose your first degree. So I'm gonna go at first with Gloria, because ladies first, sorry guys for that. And Gloria, do you mind to say me what is the main thing that made you choose biomedical sciences?
Oh, unmute yourself, Gloria.
Okay, so when I was in my A-level, I knew I wanted to do a science course, but I didn't know which one. So after doing my research, I decided to go with the broadest one I could find, because I knew that I wanted to do, even before I did a first degree, I knew I wanted to specialize in something anyway. So I thought, why not do a course that will let you know everything and then you get to choose. So that was my motivation in studying biomedical sciences, because biomedical science is so broad, it teaches you everything. All parts of science, you're going to learn about pharmacology, you learn genetics, you learn physiology, everything. So if you do that course, then you'll have quite a lot of knowledge about everything, and then from there you can decide which one you like and which one you'd like to specialize in as well. So that's how I chose.
Yeah. Can I just add to that? I think the other thing we've heard from other people, lots of people choosing biomedical sciences now. I think it is a really great degree because of what you described, but also although it's very broad, maybe in the first year and maybe second year, as you go on in the degree, you get a chance to choose specific modules. So if you are really interested in pharmacology, you choose the pharmacology ones. If you're interested in virology, you choose that. So there is sort of some specialization, isn't there?
Yeah, it's definitely a specialization, because especially during your final year, when I did my final year project, I did it in genetics. So it was good in that sense where I spent the last part of my degree doing something I really enjoyed. So after you've gone through the motions of first year, second year, learning everything, in the final year, then you get to really specialize and you get to really, really, knuckle down on something that you really like. And then from there, you can decide what you want to do afterwards, if you want to do a second degree or if you want to work. So it's all good either way.
Why did you decide to do a master's? Because a master's it's quite expensive, isn't it? So
And whilst it's expensive, but I think it's worth it, because in as much as your undergrad degree, you learn a lot of content, there's a lot of science, there's a lot of other things, but a master's will add value to your degree. It will add value to you as a person, because I know my classmates can also vouch for this, in that in our masters, we learned so much more than science. So we learn how to create startup businesses. We learn a lot of other things that will be useful to you in your future that don't necessarily have anything to do with science. So even then, a master's will give you more skills. So it will make you become a master. It literally does what it says on the tin. It will make you become a master in whatever it is that you want to do. And you gain extra knowledge, extra things, connections as well. It's really good. So I think it's really, really worth doing a master's if you want to, but I think it's worth it. I'll do it again. So yeah.
And I guess it's the translatable skills as well. So exactly what you're saying. So skills that, you know, it doesn't need to be applied to science. Like you learn how to teamwork, how to even work by yourself in your project. You learn how to analyze data, how to, you know, think differently, how to critique. So it's all these things that you're building, kind of, you're building your it's like building blocks, scaffolding. So obviously you need to choose a topic you're interested in, but definitely a master's gives you an opportunity to do that.
Yeah. And I think the other another really important point is, quite often when I go into schools, you know, people will ask me about coming to Imperial, and when I sort of say, okay, you've got to have A stars to come to Imperial, there's a lot of reason why people don't get A stars when they do their A levels. And you know, I think we recognise that. We heard from Alex, you know, he had his mind, he wasn't sort of focused on his studies at that time, and I know that's one thing. And you know, there are lots of reasons why you're not going to get A stars that don't relate to your actual true sort of aptitude and capability. And one of the things that we really look at for our students, as you've seen here, you know, we will bring students in from you know, anywhere if they've got the passion and really want to do it, because yeah, loads of us, you know, we didn't start off our sort of careers at Oxford or Cambridge or anything like that, but people can still end up at Imperial, and it's one of the best places to do science. So yeah.
And as a teacher, sorry to interrupt, someone who teaches on, you know, master's courses and at Imperial, it's so refreshing to see people coming from everywhere, you know, from different backgrounds, from, you know, different countries. It's really nice to see people, and really we can share from each other. So I think, you know, it's nice when you go to a university like Imperial, where you do have people from different backgrounds and sharing from each other.
Yeah, I mean, I'm just quite, you know, for the master's students, I mean, when I did my degree, it was all the people were from the UK. So it's great now that you actually get to meet all these people. You've got plenty of places to go and stay when we can actually fly again. Anyway, carry on. Who are we going to hear from next? Yanis or Alex? You're going to tell us about your...
Yeah, I can. Well, I was really curious when I was younger, and always attracted to these health studies, like how the body functions, how does it work, how things work, why do you get sick, how do you get back healthy. So yeah, I intended medical studies, and it was like a contest in France, which I tried twice, and well, I failed, but it's okay. Bounced back and did the biomedical science degree in the same university, and like that was more broad. So like you were studying math, physics, chemistry, biology, that all applied to health studies. Mm-hmm. It was really great to see kind of the more aspects of the field. And yeah, after that, had the opportunity to come to Imperial, and that's where I found out that you really have to be interested and curious about what you're studying in order to succeed. Mm-hmm. And like, it's a good thing. Like for me, who's a curious person, yeah, it was a good thing going after class to look up more papers on the subjects. Oh, this come out this is a new technique to treat this disease, and and that's also one thing I was taught about this year during my master's.
Cool. Yeah, that's good. Right, Alex. So, any other comments?
I honestly, I don't think I have much to add to those things. I picked the degree. I have the attention span of a 5-year-old child, and honestly, you've got to pick something that intrigues you. It was certainly the breadth, like Gloria said, of my degree, and certainly there was at my university there was once you'd completed your 3 years, if you wanted to go and study medicine after, there was a fast track program which skipped the first year of medicine. That was not attractive for me. It's not something I've ever been interested in. But certainly lots of my peers studied the biomedical science as a taster. And if they loved it, they could go on to medicine. But if not, they could leave quite happily with a good degree.
Yeah, and it's interesting to see the sort of range of things that people do, you know, after their degrees and after their master's courses. You know, we as you say, going into patenting, going into work for companies in the city and things like that, so they might just be using their skill set, their analytical skill set, in another context. But yeah, great. Okay, Margarita, is that all in terms of questions?
Yes, I think our invited speakers today were pretty clear, so it was the only question that we have.
Okay, great. Okay, well, thank you. That just leaves me to say thank you to everybody. So thank you, Blerina, Gloria, Alex, Yanis, for your talks. And that was really excellent overview. Thank you, Margarita, again for being with us, and Sam for producing the session. So we're going to be back on Thursday, this Thursday at 2 o'clock, and we're going to hear from Dr. Anna Blackney and her colleagues and how they've adopted a really unusual strategy to develop a vaccine against COVID. So these are the scientists that you might have heard about. They've been working in Professor Robin Shattock's lab, and they've been working since January. So since the COVID-19 was first identified in China, they have been working to fast track the generation and creation of a new vaccine. And they, as I say, they're using a very sort of non-traditional technique to do this. And this vaccine now, they've actually produced it, they've tested it in animals, they've had really positive results from animal studies, and so now they are just started testing in healthy volunteers. So this is obviously really exciting and important work. We've all heard many times over the past months that we need to get a vaccine for us to be able to get back to normal. So this is obviously critical. And what's going to be really exciting is that we've actually filmed Anna and her colleagues in the lab over the last sort of month, and we filmed them in the process of making the vaccine. So you're actually going to be able to see how they make the vaccine. And I think it's really, again, really interesting because, for example, Anna is a chemical engineer, but she's now working in a virology lab. So again, it's this idea that you don't have to stay exactly in your same discipline. You can move and work in and take your skills and work in another lab. And that can lead to some sort of really exciting and innovative science. So thank you so much for joining us today. There's a survey in the chat on YouTube, so please fill that in for us. Enjoy the rest of your day. Hope to catch up with you tomorrow. Please let other people know that these lessons exist. Goodbye from us now. Goodbye.
Bye, thank you.